Assalamualaikum warahmatullahi wabarakatuh. We are from group 4A3. Today we will present on the topic quorum sensing in bacteria.
Assalamualaikum warahmatullahi wabarakatuh. My name is Cik Nurini binti Latif Syam, and today our group will be presenting about quorum sensing in bacteria. To start off, there are several main molecular mechanisms of tolerance and bacterial persistence, with quorum sensing being one of them. Quorum sensing is a cell-to-cell communication mechanism between bacteria. It could happen within the same species or between different species, where they could exchange information regarding changes in the bacterial cell population density to regulate the gene expression. Quorum sensing bacteria produces and releases small, diffusible and chemical signaling molecules termed autoinducers. Autoinducers will accumulate in the environment as the population density increases. Then the quorum sensing bacteria will correspondingly adjust the gene expression. Through autoinducers, diverse but specific physiological processes like the biofilm formation, production of secondary metabolites like antibiotics, virulence factor expression, stress tolerance to biotic or abiotic factor, and stress adaptation mechanisms such as secretion system for interbacterial competition could be controlled in accordance to the fluctuations of population density. Secretion system are components to secrete virulence factor, mainly in protein form, into host cells or environment, and these secretion systems play a crucial role in the communication mechanism between bacteria.
The most commonly studied quorum sensing system is that of the bioluminescent marine bacterium Bifidobacterium. Light emission is tightly correlated with the cell population density of the host, and this phenomenon is controlled by quorum sensing. To produce bioluminescence, look I produces an autoinducer molecule called acyl homosyl lactones as a chemical signal molecule in quorum sensing. The autoinducer concentration increases with increasing cell population density. As the concentration of autoinducer increases, the autoinducer binds with loop-R protein. The loop-R autoinducer complex then binds with loop-I-CDABE promoter to activate the gene expression and generates luminescence.
For mechanism in gram-positive bacteria, firstly, the autoinducer peptide will be encoded by the AGRD. AGRD is a specific gene for the autoinducer peptide encodings. This AIP then will be released or secreted via the AGRB. AGRB is actually an ATP binding cassette. When they secreted and the certain concentrations of threshold is achieved, this will activate the AGRC or histidine kinase protein, and then eventually this will set off phosphorylations. The phosphorylations will activate the intracellular regular protein or AGRE, and will eventually regulate the transcription of the AGR genes in RNA2, which is the previously about the AGRD for transcription AIP, or it will also lead to a certain expression such as virulence in Staphylococcus aureus.
Next, I will explain about the biosensors that under the part of application of quorum sensing. Pathogen diagnostic and therapeutic is the most of the whole cells quorum sensing biosensors that have been described so far. Recognize gram-negative acyl homosyl lactone or AHL. AHL receives transcriptional regulator and a coordinate promoter which direct the transcription of a reporter gene. Quorum sensing signals alone can be used as markers for the presence of pathogenic bacteria and can be detected using autoinducers in contaminated groundwater, dairy, and milk product. For example, pathogen EC is estimated to cause more than 70,000 illness and 60 fatalities each year. Genetic circuits could be designed to recognize pathogen cell surface antigens and convert the information of cell pathogen from signal quorum sensing to a readable output. Another application is cancer detection, where synthetic genetic circuit can identify the input such as cancer surface antigen, cancer microenvironment specific condition such as hypoxia, and the biosensor's local cells density. The key requirement of such biosensors will be specific that clear differentiations between tumors and healthy cells. Microbial biosensors can be designed to recognize cancer cell aggregation in vivo. One concern in designing cancer biosensors will be to ensure that engineered microbe itself does not trigger off a host immune response or cause other harm. For example, the quorum sensing signal 3-oxocarbon-12 HSL of Pseudomonas aeruginosa inhibits proliferation and induces apoptosis in human breast cancer cell lines. This bacteria also signaling is a good starting point for developing synthetic AHLs homologous that retain anti-cancer toxicity and detection of cancer.
The application of quorum sensing in bacteria, one way I would talk about is reduction of biofouling. Now, what is biofouling? Biofouling is an undesired accumulation of microbes that will lead to contamination, colonization, and also corrosion to a machine part that been exposed to water that will lead to decrease in efficiency due to the secretion of this living organism. Now, Pseudomonas aeruginosa, Staphylococcus aureus, Staphylococcus epidermis are opportunistic pathogen that will form biofilm on medical implant that will become virulent. We can see an example of quorum sensing being applied in reduction of biofilm is Pseudomonas aeruginosa will form a thinner biofilm when culture is treated with synthetic furanol, causing in reduction of virulence factor, which is elastase and chitinase expression. The next application of quorum sensing in bacteria is biocontrol strategy. The disturbance of quorum sensing based regulatory circuit show to be effective prevention of disease outbreak caused by microbes. Inhibition of signal molecule biosynthesis using S-adenosine methionine as a substrate to block LAS-I expression, and the use of QS antagonists are two of the strategies that use auto-inducer to disrupt quorum sensing. The degradation of AHLs could be used as both a curative and preventive biocontrol technique.
Moving on to the pros and cons of quorum sensing in bacteria. The first advantage of quorum sensing is that it allows communication and alter the gene expression in response to the presence of other bacteria to improve access to nutrients, promotes defense against predation or phagocytosis, and persists in a low nutrient environment. It also resists toxic chemical. For example, selective chemical inhibition of accessory gene regulator, quorum sensing in Staphylococcus aureus, promotes host defense with minimal impact on resistance. Other than that, quorum sensing also allows a population of bacteria to coordinate global behavior and act as a multicellular unit. At a low density of bacteria, the auto-inducer diffuse away from bacteria, and there are insufficient quantities of this molecule to activate the quorum sensing genes that enable the bacteria to act as a population. As a result, the bacteria behave as individual, single-cell organisms. Acting as individual organisms may enable a low density of bacteria to gain a better foothold in their new environment. In other way, in a high bacterial density, they will behave as a multicellular population. Individual bacteria within a group are able to benefit from the activity of the entire group. As the entire population of bacteria simultaneously turn on their virulent genes, the body's immune systems are much less likely to have enough time to counter those virulence factors before harm is done. Last, but for the advantage of quorum sensing is that it could enhance pathogenicity and evade host defense by delaying the production of tissue-damaging virulence factor until sufficient bacteria have amassed and are prepared to overwhelm host defense mechanism and establish infection. The cons of quorum sensing in bacteria is that there are a significant rise in resistance to antibiotic and host immune system. A study shows that clinicians who deal with biofilm-associated infections, such as medical devices or implant infections, are frequently fail to cure their patients by using antibiotic as their treatment. There are also increasing evidence that biofilm infection often resist to the highest deliverable level of antibiotics. The formation of bacteria biofilm has been recognized as a crucial pathogenic characteristic in various chronic infections. Some example of biofilm infection is infections of bone, gastrointestinal tract, eye, urogenital tract, implant, and dental devices, and many more. They are resistant to the highest deliverable antibiotic, resulting in significant tissue damage and long-term suffering of patients. In the context of antibiotic resistance, studies of microbial biofilms led to a realization where bacteria in biofilm exhibit traits that differ from their planktonic lifestyle as they function as a group for coordinated activities. Thus, This become a problem because an effective therapeutic strategy to prevent or treat biofilm infections are currently not available due to the different group behavior. Therefore, understanding the bacterial social behavior and molecular mechanism in the development of biofilms will facilitate the development of prevention or treatment of biofilm infections.